the only date that we could not get the biotech place for, we have to go to the PTCRC. There's a large room as you walk in that lobby, off to your left, called the Chestnut Room. So the security guard already knows that you're going to be coming, so you probably don't need to sign in. She'll just point you in that direction. So we look forward to having Ronnie Matry come. He's an adjunct professor with the Department of Physiology and Pharmacology. He's worked a lot with the folks in the cardiovascular research, and he has a good deal of interest in the ADME principles that we're going to learn a little bit about today. That is absorption, distribution, metabolism, and elimination. So the general structure of our course, even though we're going to be covering topics that are fairly widely distributed, is we're going to begin starting with how drugs get into the body, how they are distributed throughout the body, how the liver takes a major role in metabolism but is not the only organ, a little bit about how drugs are removed by the kidney, some pharmacogenomics, which is an up-and-coming field, and most drug companies are putting a lot of effort now into figuring out how the genome regulates particularly those kinds of enzymes that are in the liver. So we have a section on that. Then the middle section of the course is on what we call classical pharmacodynamics. That's how the cells respond to various kinds of drugs. And we're going to put a major effort on those compounds that are working through G protein coupled receptors as well as transporters. And then we're going to go a little bit deeper after we've gotten through the receptors and how they work into what some of the signaling pathways are that are in cells. Most of what we see in common here is that they all work by phosphorylating key substrates and dephosphorylating them in order to change the behavior of the cells. So we have two kinds of learning experiences, one of which is going to be your same old lecture. That would be an hour and a half of listening to someone else tell you what they know. But then we're going to go even further into what's known in the rest of the world. And that's where we rely on our research literature, is what we call it. It's not the same as reading poetry. It is reading journal articles. And we're going to ask you to present journal articles to your group. And we're going to be doing those on Fridays. So you can see that for the most part, for the rest of the Fridays, we're continuing on. Now I want to point out that on Fridays, we're going to begin a little bit earlier in the day for several reasons. One is that there isn't an MCB class that precedes things on Friday, so you can actually come to class a little bit earlier. Number two, there is a seminar that some of the people in here may be wanting to go to for molecular and cell biology that begins at 11. So we've adjusted some time so that people can adjust. If you don't go to the seminars at the end of the day, you can prepare for your grad 713s, I guess. Now, we also have too many people for everyone to be able to be in the same group. So I have divided the group up into two so that we can have one group, Group A, which everyone has in common of being interested in a clinical career. So the people who are going to be working with your group are going to be choosing papers that are more translational and clinical in orientation. Group B, on the other hand, are people who have an interest in a research career. And so they're going to be spending much more effort on papers that involve basic science bench research, the techniques, how the techniques are used, and have discussions around that. So because of that, we've got some additional people that are going to be working with your groups. so that if you do need to contact anyone at all, you've got their email addresses, and you've also got a little bit about their credentials, what departments they're in and where they're from. So you'll be able to do that, and the people that are going to be working with the clinical groups include, I'm gonna start, but then we're going to be moving into Bitta Nickel and Iris Lang, who both are MD PhDs. So they're going to be choosing the papers for that group when it gets to be their turn. Dr. Shaltu and I will be taking the research group when it gets to be related to the kinds of things that we're going to be talking about. And we also have Dr. Petrovec, who is an MD, who's going to begin working with either group, but I think predominantly the clinical group. And then the research group is going to have one time with the speaker, Ron Chen. So the way this is going to work is that those faculty members are going to choose the papers for the group to go over, and a week ahead of time, they're going to email those papers to the speakers. So in the clinical group, we have twice as many people, so that Friday morning, 2 different people will be going over 2 different papers and present that to the rest of their group. And you're not going to be sitting in your group not doing anything but listening to them. You're going to be having the papers on Canvas ahead of time so that you can look them over. If you have any questions, you'll be able to ask those and make it more of an interactive discussion, because this is really a good opportunity for you to try to understand what's going on in research labs in the rest of the world, and you can't do that if you have a question and you don't ask it. Group B has fewer people, so that will be 1 person with 1 paper per group, and that could easily generate more discussion. So you'll be getting these emailed to you from the faculty member who's in charge of that particular date, and they're all listed here, so that you can begin presenting your papers. Now, I think most of you have had some opportunity in presenting in journal clubs or other activities, is that right? Has anybody totally never presented a research paper before? Okay, good. Well, you know that you want to begin with some background so that your colleagues all understand what the importance is and why this particular set of studies was important to do. And then you want to, if it's one major kind of a method, then you want to tell us a little bit about that method. But in research papers, when you read them, all the methods are in one section together. If you present it that way to your group, then chances are they're going to have forgotten what you told them by the time you get to the point of each one of the experiments. So what I would recommend you do is for every figure that's in the paper, and for most of these you can download them directly from the website. Don't try to take them off of the PDF. You may choose to get the PDF so you can read it and it looks neater, but if you go to the original article and you click on these figures, sometimes they'll even give them to you already as a slide. Otherwise, you can copy and paste the figure, and that gives you much higher resolution. So what I would do is to start out with each and every one of the figures and say, first of all, why they did the experiment. Then the question would be how they did the experiment, and that's where you can go back to the methods so that you can describe what was done to actually perform that experiment. And then what were the results, and that's where you say this went up and that went down. More importantly is describing why that result was important and what it really means with respect to why that experiment was done. So that often leads into what was now figure 2. Figure 2, why did they do this experiment? Maybe it was because they found something to some of the websites like Amazon.com or Barnes & Noble, chances are you can find somebody who's got used textbooks. And unless the book was just published last year, you'll probably have lots of opportunities to get used textbooks for much less. And if you don't like them at the end of the semester, you can always put them back on the market and sell them again. So, or you can begin keeping your set of textbooks. But we've got basic and clinical pharmacology. This is a textbook that's easy to read. It is the kind of thing that folks use when they want to study for boards, and it's part of a series of similar kinds of review books. So it doesn't have a great deal of information, but it's got all of the main focus, all of the right vocabulary, and the kinds of things that you need if you don't have a lot of time. Now, for those of you who are going to be going into this field as a profession, you might be more interested in reading through Goodman and Gilman's The Pharmacological Basis of Therapeutics. This is the Bible of pharmacology. This is what you see when you go into any clinician's desk, or a veterinarian or a pharmacist. They all rely on this largely as a tool for getting more detailed information. So it can be used as a reference book. But if this is going to be your primary career, or if you're interested in cancer drugs, and we're not covering those very well, this is really where you want to go for most of your detailed information. For those who are going to be looking a little bit more at the physiology, and some of our lectures, such as those on the kidney, may not cover physiology in the way that you would really understand it if you didn't have a good undergraduate physiology course coming in. So Guyton and Hall is a very good physiology book. It's in our library. Recently, there's been a newer medical physiology book by Boron and Volpe. That goes into great detail, maybe a little bit more detail than what you want, but it's a very excellent book. And it is also available. When we come to the signal transduction, that means beginning with what are receptors, how do receptors work. We have a signal transduction book that's the second edition, and there's one that's the third edition. The third edition is maybe five chapters that are very different from the ones in the second edition. It's like one of the authors decided that there were new things that were signal transduction pathways hadn't been covered very well in the second edition. So they're really two different kinds of books. The one that I'm assigning most of the reading for is the one that's in the second edition. And my understanding is that if you click on this, you're going to get the second edition. And it's kind of hidden away in the library archives of what we have online. Whereas if you click on some other pathway, you may get the third edition. So the third edition is not going to be one that we're going to be studying from, but the first two chapters can give you a synopsis of what is in the first, the second edition books. So you could probably read through those two chapters. So I've given you several options here on reading. So we've got two columns here. For those who want to start out with one chapter and then maybe go to something that's a little bit more advanced, the second column is more advanced. Then finally, we do have to evaluate your learning process, and the best way to do that is through exams and homework. So we're going to be giving you homework from time to time, and where is the homework? And then the exams are 3 exams. So the 3 exams are going to count 50% The student presentations on Friday are going to count 10%, and the homework is going to count 40%. This is the grading rubric at the end of the course. During the course, you're probably not going to be able to calculate exactly where you may fall on this, but we are going to be including A minuses and B pluses and B minuses, so it's not just going to be an A, B, C delineation. And that's the way the graduate school is looking at these now. I've got some academic policies that essentially say that cheating is going to be a no-no, but of course you knew that already. So the other thing is learning objectives. We've got a lot of lecturers. Most lecturers are going to start out with some kind of learning objectives, but you all have different career goals. So you're going to have to think about every tidbit of information you get as to how you can put that into your career goals and help you to make it stick in your brain in a way that your brain functions. And everybody's brain functions a little bit differently. The way you file things in file cabinets. So you have to begin thinking about how you're going to be assessing the information that you're given and how you can utilize that best in your career. And so at this point, you're going to have to begin thinking a little bit about defining your own learning objectives, because a lecturer can't say you need to know this, you need to know that, you need to know this. Because maybe that's not what you need to know for your career. So you have to begin assessing this yourself. So I've given you some guidelines here, and it involves working with some of your colleagues who may offer some advice to you as well. So does anybody have any questions at this point on what we're going to do? Yes?
If you please could email us the presentation 2 weeks before, because we have other presentation, we might have exams, so that help us prepare better.
Could be. I can check with the lecturers, but if you have something that you know in advance is going to conflict with what you need to do in another course, my understanding was MCB doesn't have research reviews, but if you want to come to me in advance and ask me if we can switch you with somebody else, we might be able to negotiate that. So I think that's better than asking for something 2 weeks in advance. Everybody else is going to have 1 week in advance. So, yes?
When are we going to have access to the PowerPoint on Canvas?
Right now. So I think it was loaded up last night.
Oh, I don't think the site has been published for your class.
Yeah, it's not active.
It's not active?
It's not published.
Oh. Okay. Well, I will check to make sure that happens as soon as our class is over, and I can email the Canvas master. Okay? Alrighty. Well, are we ready to begin? So today we're going to be talking about some general principles in drug discovery, design, development, and distribution, and how drug companies begin to work with this. And we're going to start with some history. Pharmacology is one of the newest fields or disciplines of investigation, and it tended to actually begin to appear at about the time that training of physicians actually went through medical schools. Prior to the 1800s, most physician training was by an apprenticeship, but medical schools began forming, and the first pharmacology professor was working in the early 1900s, and his contributions included isolating adrenaline, and we can think of adrenaline or epinephrine as the first neurotransmitter, but also were born with type 1 diabetes and needed to have insulin replacements. Does anybody know why we use epinephrine right now? What epinephrine is used for? Anaphylaxis, yeah. And if you pay some attention to some of the websites that, I don't know how I ended up getting a lot of these websites, but they're the ones that want me to sign petitions. There are a lot of parents that are signing petitions right now because the company that produces the epinephrine EpiPen, which their children need if they go out and get stung by a bee, has vastly increased the price of that drug, and to the point where some of them feel like they're not going to be able to afford this for their children if they need it. So, very important drug from that point of view. Now, historically, drugs were not always under the same kinds of protections that we have now here in the United States or in Europe, and we're going to be going over a little bit of history at the beginning here now on how we've gotten protections, because that helps us to understand what drug companies, the pharmaceutical industry, needs to do in order to provide you with those protections. Before the 1800s, and largely I think we're demarcating at a point that was the war between the states here in the United States as one of the landmarks of when pharmacology really changed and the ability to distribute drugs. Prior to that, we had anesthetics like alcohol, but sometimes that was only one of several ways. If there was going to be any kind of surgery, usually you had to hire heavy guys to hold somebody down, as well as give them the only anesthetic that was available, that would be alcohol. So we've moved now to the middle of the 1800s, where the hypodermic needle was first invented. And that was about the time when morphine was becoming to be a very prominent analgesic painkiller for the soldiers that were in the war between the states. And for those of you from New Jersey and upstate New York, here in the South, it's called the War of Northern Aggression, in case you didn't catch that. So here we've got several aspects here. Once the hypodermic needle was invented, then you could begin injecting drugs, such as morphine. But we still have our natural products, and morphine, of course, has come from opium poppies. Cocaine comes from coca leaves, and these were widely distributed as the kinds of tools that could be used for pain relief. So here we've got toothache drops that include the anesthetizing actions of cocaine. Now, at some point in time, we realized that simply taking things from plants is not going to be the best ideal. Sometimes they're too toxic, sometimes they don't get dissolved in teas or elixirs, sometimes they have to be modified. And that's what drug companies do, is they take what is the known active compound from a plant or another natural product, modify it so it's a better drug. And one of the first companies to do that was based on work from a Professor Bayer, who realized that taking salicylic acid from some of its natural sources, which include willow bark and spirea bushes, was extremely toxic to the GI tract. But he realized that when he mixed it with acetic acid or vinegar, he'd be able to acetylate that salicylic acid. We have acetyl salicylic acid now, and that's aspirin. And it really revolutionized the ability to use this as a painkiller and an anti-inflammatory, because now this kind of toxicity to the GI tract was reduced. It was not eliminated, because we know now that these compounds work through prostaglandin inhibition, and that's very important for protection of the gut. But at least in the ingestion process, the absorption process, that could help. Now, he took his method with some of these soldiers who were beginning to get morphine addiction when they were no longer needing the pain-killing parts of morphine and they continued to use it. They were developing addictions, or they were developing tolerance, which means that you would need more drug to get the same effect. So we thought, well, you know what? I acetylated salicylates and it worked. Let's acetylate morphine. And he got heroin. And it was originally advertised as, well, and of course it's used here as a sedative and for coughs, but he was advertising it as an addiction-free morphine analog. Now, is that what we know now to be the case? Probably not. But, you know, different countries have different rules and regulations. In Europe and Great Britain, heroin is used, but here as an analgesic, but in America it's considered to have a high risk of addiction, so it's not. Now, one of the problems as medicines began to be dispensed, not only from plant products, but from things that are now going to be put into what's called patent medicines or extracts that are bottled, was that we really didn't know what was in them, and they could be sold without having any kind of indications. They could be given as to what they were needed for, but the actual dosing regimens weren't identified, and safety wasn't really identified or even tested. So we have things like cigars of joy that were advertised for use in asthma and bronchitis. I have a feeling I know what cigars of joy are, but you know, I don't know that for sure because it doesn't tell me here on this ad what it is. Does anybody think they could guess what a cigar of joy might be?
Is it legal in Colorado?
It might be. That's what I'm guessing this is, but of course you can see that you don't know. So here we've got a question of safety. So we don't know whether this is safe or not, but it's probably very effective for a nursing mother to be able to ingest alcohol, which will come out in mother's milk and maybe help put the baby to sleep. That could be effective, but is it safe? And we really don't know that. Another thing, you know, right now we're battling in our country and throughout the world an obesity problem, which can lead to cardiovascular disease. But, you know, this is not new. This is an ad from, I don't see a date on here, but it's clearly early 1900s, a mechanism of banishing fat. And, you know, no ill effects. It's easy to swallow. It's doesn't require diets or baths or any kind of exercise. Tapeworms packed in a jar. Now, is that safe? Was there any kind of regulation at this point that suggested that, you know, it's probably, again, very effective, but is it safe? So beginning in 1906, in our country, our legislature in the federal government established the Food and Drug Administration, and they did this through an act of Congress, which was the 1906 Federal Pure Food and Drug Act. So right up until this time, there was no kind of regulation, federal or otherwise, on the drugs that we were able to use, or where we could get them, or how they could be sold, or what they were used for, or whether they had any kind of side effects that needed to be known. So the Pure Food and Drug Act had three main requirements. First of all, it said that the foods and drugs must be pure. So that means that blender or smoked or something, and we don't know what the contents are, then they're not pure. Second, you have to have the labeling. You have to know, according to the U.S. Pharmacopoeia, which is what USP stands for, what is in that drug. And then the Food and Drug Administration was established in order to be the arm of the federal government that would monitor what pharmaceutical companies were able to sell, what their labels looked like, and what they were able to say that their drug could be used for. Well, it wasn't until 1938 that we included one more addition to this, and that was through the Food, Drug, and Cosmetic Act of 1938, that the drugs actually had to be safe. So what this means is the Food and Drug Administration had to monitor the studies that were done by the pharmaceutical company to demonstrate that they had been tested for toxicity and that we knew what some of the toxic side effects were. So this was not until 1938. Then in 1962, another act of Congress, the Kefauver-Harris Amendment to the Pure Food and Drug Act, was that drug efficacy must be demonstrated. Now, efficacy is a term that we use for pharmacology, meaning that the drug is effective. And so your question has to be, well, what is it effective for? That's where the pharmaceutical industry has to demonstrate that there are a certain disease that this drug is going to be useful for treating and provide the evidence for that. Largely that evidence is going to come from not only basic science research, but from clinical trials. And we'll talk a little bit about what those clinical trials are in a few minutes. Efficacy at the molecular level means how well does the drug work at the cell level and what it does. But efficacy here was meaning how effective is it and what the drug company tells you it's going to do. Sometimes a physician will use something, what we call off-label. That is to say, they say, well, you know, the label says that it's very good for treating epilepsy. But it does that through the glutamate receptor, and now we've got drug addiction, and that also involves the glutamate receptor, so let me try this drug, which the drug company proved was good for treating epilepsy. Let me try it on these patients over here. That's called off-label use. And that really takes the level of responsibility away from the drug company itself, because they didn't approve it to do that. They took it to the Food and Drug Administration saying it was going to be useful for this, that, or the other. Yeah.
Is that legal still? Off-label?
Yeah. Nowadays, if you look under clinicaltrials.com, you'll see a lot of people that are putting these drugs through their own clinical trials for what they think might be a very good idea.
And that's independent of the drug company?
It could be, or it could be sponsored by the drug company. So, right now, it's a lot easier to take a drug that's already on the market and repurpose it for something else, because it's already been tested for safety, and it's already packaged in a way that the Food and Drug Administration approved of it. So it's a lot easier for someone to take a drug that's already been approved and say, I wonder if it works this way, and put a clinical trial together.
And would that renew the copyright if it's found to be effective for something else?
Well, it's not a copyright, it's a patent. And the patent is something that the company that produced it originally would hold for a limited number of years. So it could be yes or it could be no. Okay, so finally, now in 1962 I was alive. None of you were alive, right, except Okay, so I'm the elderly person here. I just cannot believe when I think about this that it wasn't until I was a child that we actually had some safety requirements and some efficacy requirements for our drugs. But I do know that at some point in time, some of the advertisements that they had for so-and-so's little liver pills and things like that, they just disappeared. So, you know, this was effective. If you can't demonstrate that Carter's Little Liver Pills was going to be good for your liver, then you could not put it on the market. So some things are on the market, even though at this point they may not be as safe as other things. Aspirin. If aspirin would have to go through the safety requirements now that other drugs do, it may not have been approved by the Food and Drug Administration. But the thing was, it wasn't until you all were born and were children, or young adults, or however old you are, that the Agricultural, Rural Development, and Food and Drug Administration Appropriations Act, that means that had to do with money that was being dispensed in the budget of our government, said that pharmaceutical companies needed to provide information on side effects to consumers. And that just happened to coincide with a period of time in our lives when computers were being used. So now, what this means is that it's not just the doctor that looks up in the physician's desk reference to find out what those side effects are. Any consumer can look up and see what side effects are, what drugs are used for, a little bit about the dosing, and other little tidbits of information, including if the drug was having some concerns that might take it off the market. In fact, anybody can Twitter and tweet and send emails and put things on Facebook that say, I took a certain drug to block my smoking so that I wouldn't have to smoke cigarettes anymore, and oh my God, it just made me drove me crazy. You know, once you put these kinds of things on a website, everybody can see that. So, you know, it can go a little bit further, but this is where the pharmaceutical companies are responsible for providing information. And interestingly enough, it was packaged into a bill that our legislature would vote on that had to do with appropriations for agriculture. Strange place to do that, but that's how our government gets things done. Now, we've talked a lot about Food and Drug Administration. It's their responsibility now to make sure that drugs are effective, that drugs are safe, that the information on the package label is correct, and the package label is going to be in a compendium called the Physician's Desk Reference. And you can get that online. You can get what is a sort of a synopsis Physician's Desk Reference online. But if you go a little bit, dig a little deeper, you can get all of the information that you're going to need when I give you your homework assignment. Or all the information you're going to need if you ever need to look up a certain kind of a drug, because that's what is put on that package insert. Okay, now how do drug companies generally make their drugs? They start out with natural products, and traditionally that's been the way that drug companies have worked right up until we started having a revolution in two things. One, the way chemistry is done so that you could make series of lots of kinds of analogs of compounds. And two, computers that can do simulations of proteins so that you can actually look to see on a computer screen where your drug might bind inside that protein. So with those two, it's really changed the way that drug companies have started developing drugs. But right up until the late 1990s, 2000 or so, most drug companies started by finding out what folklore medicine was using, purifying and extracting what the chemical compound was in that natural product. And the natural product didn't need to be necessarily a plant. Snake venoms, spider toxins, all of these, mushrooms are very good sources of frog skin, all those things that the Wicked Witches were using. Very good pharmacologists, those Wicked Witches, and they knew what to do with those things. So drug companies would take them. Drug companies are really concerned about the plants diminishing in certain parts of the world where the native cultures are using them kinds of diseases. But what we really need to pay attention to is that we're not using those plants. We're using active compounds that came from the plants. Then researchers can identify how they work in the cells. And so that's why we're giving you the pharmacology knowledge of pharmacodynamics, how the drugs work at receptors, how they change the way cells work. And then chemically modifying these so that they can increase what's called the potency. Now the potency means how much drug you need in the body in order to be effective. So if you had something from a plant, you knew what it was. If your chemist could modify it a little bit, so instead of needing 100 micrograms, you maybe now have a new chemical that only needs 1 microgram. That's what makes it more potent. That modification can be done by the chemist and the drug company. Or maybe increasing its efficacy. Maybe it doesn't work very well. Actually, THC from those plant products that you get from Colorado doesn't really work as well as some of the synthetic compounds that drug companies made for pain relief. Increasing what we call the half-life. That's another word you're going to need to know. That's how long it takes for a drug to be removed from the body from 100% down to 50%. And for most drugs, that same amount of time is going to go from 50% to 25%. And then from 25% to half of that, 12.5%. So we call that the time that it takes to diminish by a half. We call that half-life. So most of what you're going to be talking about with respect to how long the drug lasts in the body, you're going to be talking about its half-life. And Dr. Shaltu is going to be showing you a little bit about how to do that. And then stability and dosing forms, because that's what drug companies do is they package it. Now, on to the new millennium. Now we've got lots of drug companies that take a rather different approach. The way their approach is to do what we call structure-activity relationships on a series of analogs of their best drug to be able to see what chemical parts work and what chemical parts don't work. And they are going to take this series of compounds, look at them not only on computers, but also make what would be a whole matrix of modifications on their compounds in order to see what works best. So one of the things that we've got to be able to look at is that some of the drugs that really work best are those drugs that are small chemical compounds. Somebody from Pfizer developed what he called the rules of 5. And he had 5 different attributes about a chemical that indicated whether it would be a good drug or not. And one of those is the size of the drug. Another one is how lipid soluble it can be. And some of these other attributes. So that a chemist could easily begin to kind of look at a drug to see whether it's going to get into the body, distributed into the body, and then act very well at a receptor. So we also had an evolution in genomics because when the Human Genome Project was completed, we began to realize just how many proteins the body was making. And from those, some of these are proteins that are likely to be involved in diseases. And so of all the 30,000 genes that could be identified, maybe 3,000 of them could be readily identified as a protein that's involved in a disease. But by the same token, some of these proteins, or some of these identified genes that were in the human genome, weren't really good protein targets for small molecules. So perhaps of the 30,000 genes, only 3,000 of those would be what the pharmaceutical companies call druggable. That is, can you make a small molecule that can actually interact with these proteins? So when you put on your Venn diagram combining the two, of the gene targets, of the proteins that can be made, maybe 600 to 15 are going to be druggable targets. So drug companies started looking at some of these so that they can really begin to develop new chemical compounds, not those that may have come from plants that folklore medicine was good for a disease, but now these are new compounds. And this was helped out by the ability of these compounds to be able to be screened in high throughput screens. Well, there were some things that we really needed to know about each one of these chemical compounds, because a chemist can make a whole series of analogs, but they're not going to be a drug until they've actually met certain criteria. So drug companies will number their chemicals, and they now are, for most drug companies, numbered in the billions. So they've got, let's say, Pfizer started out with CP, Charles Pfizer, as the first two letters, and then they started putting numbers. And then when they started going global, after they started getting their Viagra money in, they changed it to PF. So, so you've got a whole series of these chemicals. These are not readily called drugs until they have met the criteria of safety, efficacy, and the drug company has decided to market them. So for those of us who are researchers, some of these we can take advantage of. Most of the drug companies are going to allow you to use some of their compounds, and they'll make arrangements with your organization so that patents are not overcome, that you will be able to have some donations from drug companies. And chances are they're going to be a 2-letter followed by a number, because they won't have been taken through to develop drugs yet. But what are we looking for in these druggable compounds in the marketplace? First of all, is the molecule active at its primary target? So here we're talking about potency and efficacy. And this can be screened in a lot of basic science research screens that may involve radioligand binding assays, ELISA kinds of assays. And you're going to be talking in greater detail about this a little bit later on in the series. Question about, is the molecule promiscuous? So what this really means is selectivity. You want to be able to give a drug that's going to be selective for the one protein that you want it to interact with, but you don't want it to be pretty good over here and good at 5 other places, because then you might be developing something that's going to have 5 different side effects that are unrelated to your original target. So selectivity, we usually use the word selective rather than specific, because specific means I definitely know that this compound is only going to work at this one protein target. Well, we can never ever say that, but we can say selective because we can use 1 microgram to get this effect, whereas some of these other side effects we need to have 1,000 micrograms. So we're 1,000-fold difference in our potency. So we can call that selectivity. Is the molecule toxic? So this goes back to Food and Drug Administration requirements. You have to test whether the drug is toxic. And you have to test whether it's likely to cause birth defects. Is the drug absorbed, distributed, and does it stay in the body for a reasonable amount of time? You know, it doesn't pay a drug company to put something on the market that you have to take once every hour or once every 2 hours. Birth control pills would never have been as successful as they are if you had to take 4 of them a day. So the advent of modifications on the drug so that it could avoid passage through the liver, and its modification that would make it ineffective, made it so that it could last, so that you could take 1 birth control pill a day, and now it's a useful drug. So some more rigorous things. from the point of view of the drug company, is the molecule different from previous molecules that have been made? You know, if you've got a Me Too drug and it's not really going to be any different or any better than something else that your competitor has on the market, it's not going to be useful for a drug company to try to develop that. And does it incorporate the newest knowledge of disease and pharmacology? So that's why we need to know a little bit about physiology, a little bit about how these drugs are working, and how we can begin to develop some new ideas for developing new drugs because we know more about the way things work. So from a pharmaceutical point of view, the process of drug discovery starts with chemical libraries of lots of drugs, medicinal chemists who can make new drugs, natural products where we can extract drugs, and we move over into a drug discovery phase, which is how does this drug actually work? And there our basic science knowledge becomes very, very important, because this is before you ever begin to think about clinical uses. You really have to know a little bit about how it works. And then you have to know about how it gets into the body and how it's distributed. If it doesn't have the ability to get into the brain because of a blood-brain barrier, but your disease is a brain disease, then that drug is not going to be very useful. So you need to know that. And once you've gotten that far, then the pharmaceutical company makes a decision about whether to make this drug a marketable drug. So some of the things that some people find very exciting are heme informatics. This is how does the drug work, and a little bit more about what those series of drugs are in what we call structure-activity relationships. So this really moved forward in the field when we were able to develop radioligand binding assays that we could do what we call high throughput screening. So on our little multi-well plates, we've gone from 96 well plates to 287 well plates to even more, where you can do little assays in very small amounts that can tell you, using cell culture, whether your compound is going to be effective at a certain cell event. So this means that you could actually test 10,000 compounds at a time. And the computer capabilities, these compounds can all be bar graphed or bar coded so that high throughput can actually go through the computer and spit out some names of some of these coded compounds that drug companies have as this drug, compound. 5 compounds may be useful according to this particular screen. We've got new kinds of equipment. Some modify cells that are cells in culture so that they can signal through a calcium change that turns on or turns off so you can easily see fluorescence. Now I want to let you know, I don't know whether this is going to come up or not, but there is a website that's very much like PubMed called PubChem. And if you go to PubChem, you can put in the name of any compound that you want to search, and you're going to get any of the interesting results from there. So let's just see. This is a company compound from a natural product that you can get in Colorado. I must have spelled it wrong. No, I didn't. Okay, well, maybe I need to put something else in. Okay, so this is giving us lots of papers that you can go to for annotated results, and So you can easily go to PubMed to play around with what you can find from some compounds that you may be interested in that may already be drugs or may be on the way to being marketed as drugs. Okay, so our simple screening assays, I already mentioned, generally can be found if you can modify a cell culture line so that now you've got a receptor on it that you want to be your target. You can provide many chemicals so that you could see whether we're going to get effects in the cell that are going to indicate that small molecule is actually going to work through these receptors. And sometimes you want to block the response, and that's what we call an antagonist. So an agonist is something that's going to cause the response, an antagonist is going to block it. So you need to have a cell line. Some people make their own, other people buy cell lines that are readily available, and these kinds of things can readily be done. So the drug companies will work through these early screening process and identify the compounds they think they want to work, and then they have to go to the trouble of modifying them maybe a little bit further to get better drugs. So they call that original drug something that could be a scaffold so that you can begin modifying it, putting amine groups here or methyl groups there, and get something that might be even better. And then you can compare those structures in computers called structure-activity relationship studies so that you can see what is important on each one of those molecules that might be the best for those kinds of screens, make even more drugs, and finally get something that may be the best drug you have. And this really takes a lot of effort, and if you're going to be doing these kinds of screenings, sometimes it's better to make some predictions. And this is where we've got in silico pharmacology. That means that if you can take your protein target and make a model of it on the computer, then you might be able to do your screening, if you know where your ligands are binding, by actually looking in the computer first. And that can save the problems of having to make a lot of new drugs. And many drug companies or small startups actually do this. So I've given you a list of some of these that you might want to just click on their websites, and Tripos is one of these. That is a drug company support structure. Companies will go to them and say, I've got this protein target, I want small molecules, and they'll take the trouble of running it through the computer systems and suggesting that you might want to do this, 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 and this. And now Tripos will even actually make those compounds for you that it's predicted that they're going to work. Okay. So our drug companies are going to take us to the point where now they can begin to do pharmaceutical development. And you're going to be, after we've figured out what they do, you're going to be studying a little bit more about the pharmacokinetics in this whole semester. And we really rely on your ability to understand how the drugs are absorbed into the body, distributed in the body, metabolized, and eliminated. So this is the first third of your course here. This is the kind of what we call ADME principles that every pharmaceutical company has devoted a good deal of their workforce to trying to understand, so that they really understand how that drug or whether that compound is going to actually make a good drug. And it's only then that they take it through the decision-making process as to whether to take one of their best compounds and try to develop it in the marketplace. So what we've talked about so far have been what we call preclinical studies. These all have to be done before that compound can ever be taken into a human. And so once we've gone through all of this, the drug company takes that information to the Food and Drug Administration with an application called an investigational new drug. And if the Food and Drug Administration approves that, then the company or the individuals in the And that's where we begin putting it into humans. So once the IND has been approved, then we can begin with what would be our clinical studies, which would be starting with several phases of clinical studies. Phase 1, phase 2, and phase 3. So I'm going to give you a little bit of an overview about what those are. Our preclinical studies move us into phase 1. Phase 1 is giving your compound to a small group of people, 20 to 100 is a good ballpark figure, of healthy volunteers. And it's largely to detect safety issues. And at this point, we've had so much work done in animal models that there is good reason to believe that there won't be too much problem with giving this to healthy people for an initial test. And this is where we also begin to test dosing. Now that's not to say that a major fiasco cannot happen, as happened about 3 months ago with a drug that was being tested in a site in Paris. And there were 5 healthy volunteers, and it's really not clear what happened because the testing company hasn't submitted their final reports on this yet, but 2 of them died of cerebrovascular hemorrhaging, we think. So, you know, here this was a situation of what should have been a simple test in healthy volunteers that went bad. But once this phase is over, and this is fairly simple, then the drug goes on to phase 2. Phase 2 is going to be asking the question, is this drug actually going to be useful for treatment of patients? So at this point, it's a small number of patients, but it's usually those patients who maybe are not going to be able to be treated by the current drugs that are available. So at this point, you can compare what they're doing with the fact that these people may not have been well treated with other kinds of drugs. So again, it's a small group, maybe 100 to 500 patients done in several different clinical labs and just tested. So if this was a cancer treatment drug, for example, these people might be fairly well along in their disease because maybe they've tried chemotherapy, maybe they've tried antibodies, maybe they've tried some other things that haven't been useful. If this new drug gives you the opportunity to think that maybe this drug is going to be useful, then it will go on to what we call phase 3 clinical trials, which are people in the thousands. And usually those kinds of trials are done in major medical centers and more than just one. So if you're going to be aiming to get 3,000 people, you may contact 10 different major medical centers and ask each one to enroll 5 to 20 patients each. And that way you'll be able to get your patients. And at this point, there's a good deal of effort done to make sure that the drug dosage that you've assessed from phase 1 is safe and phase 2 is useful. And now you're looking for what would be a broader scope of side effects as well as whether the drug is actually working. Now at this point, this drug may now be sent back to the Food and Drug Administration with all of those data. And then phase 4 is after the drug company has put it on the market. Because at that point now, you've got a much broader range of people. And the drug company is still responsible for reporting any kind of incidences of drug safety or toxicity. And we call this the post-marketing or phase 4 effects of the drug. Now I want to point out that of all those chemicals that a company may make, and they may have identified 10,000 chemicals that they run through high throughput screens. Maybe they take 250 of those chemicals to try to be able to go through the basic science studies, preclinical studies, but they're only probably going to think about getting 5 of them that they seriously consider to go through any kind of produce the drug.
Well, can they, okay, so they can't be like different, they can't be like too different in their mechanism of the like the compounds of the drugs? It has to be pretty similar each one?
Well, what you're doing is narrowing down so the drug company can get its best drug. So, and there may be more than one drug company working on something, so there could be analogs that could come on the market. Now, I want to take just a minute to tell you about how you would do this if you were going to do this here, and this would be through a clinical trial investigation that would have to be approved by what's called our Institutional Review Board. And the reason is that we are very conscious about human subjects research as being requiring that the person be able to make up their mind about whether they want to do something or not. And this is called the principle of autonomy, so you have to provide informed consent. This would be a written document that gives the volunteers all of the information that you know that would be useful for them to make a decision. You have to explain to them what the benefits of being in a clinical trial would be. Is this drug potentially going to be useful for you and your disease? Or maybe it's going to be useful not for you, but maybe all of the other people that may follow you might be a benefit. And then the first do no harm. If you're going to have side effects that you might predict would be there, you're going to have to explain that to the patient when they volunteer for a clinical trial. Principle of justice. This has taken on some new effects in recent years because you can't really deny somebody's opportunity to be in a clinical trial, but very often clinical trials were kind of limited to one group of people. So a lot of clinical trials that may be involving cardiovascular disease drugs were taking some of the main sources of cardiovascular disease, which were men, totally ignoring the fact that women may also have heart attacks as well. But now you are really bound to take people from different kinds of genetic and ethnic and social groups. But on the other hand, you can't be taking people that are in a population that can't make these decisions themselves. So at some point in time, people were taking advantage of incarcerated people from prisons. And here you've got people that are totally under your control, but you really can't do that. So you can't be taking people who can't make a decision. So that would be people who might not have the mental capacity to make a decision. So all of that falls into this principle of justice. And then conflict of interest. Here at our institution, as well as many other institutions, we have people who are taking drug company money to be consultants. If they were to now enter into a clinical trial where they would be giving drugs to people while they're taking money from a drug company to study that drug, that becomes a potential conflict of interest. So with all of these things in mind, we have to actually do our clinical trials in a biological way. Otherwise, we may have some problems. So how do you know where clinical trials are being offered? There's a website. When you do a clinical trial, you register with clinicaltrials.gov, and you can go to that website. You could look for any kind of description of a disease, or you could look for a drug itself and see who's doing clinical trials on those. And that's going to be part of your homework. So your homework is the very last slide on this slide set, but I do want to tell you a little bit about naming drugs before we go over the homework. Some drugs are what we call over-the-counter, OTC. Those would be drugs that have been already used by vast populations of people that are considered to be relatively safe, where the consumer can make the decision on whether to give themselves the drug or give their children the drug. On On the other hand, many of our drugs are prescription-based and have to be dispensed by a licensed provider. So those are the ones that have been through the Food and Drug Administration, and the Food and Drug Administration has said, no, we're not going to make this over the counter. You have got to have this under some kind of control. Now, we already talked about when the drug company makes these, they cannot possibly give you a name for virtually every chemical compound they make. So they start out, first of all, chemists will know those long, long names that you've all seen that have the words ethyl and phenyl and dipropyl and yada, yada, yada in them. They're not very useful for us, except when we publish papers on them. That's what we need to tell people so that they can know what that compound is. But generally, the drug companies are going to give them a code name, and then if they decide to take them through these clinical trials, they're going to give them what's called a generic name, and that's where the drug company has put a name together, and that's going to be its non-brand name. So it has to have some official approval, and that's what we call the non-proprietary or the official name. And virtually anybody who was making this compound would be calling it by that name now. A brand name, on the other hand, would be what that company is going to sell it by. So it could sell it by the name of Advil, but that's not going to be its generic name. That's going to be the brand name that was chosen for the marketing purposes. And chances are they're going to hire a marketing company to help make up that name so it could be useful. Now, the names of the generic drugs are going to be approved by a council that includes representatives from the American Medical Association, the United States Pharmacopoeia, and the American Pharmacists Society, or associations. Our generic drugs may contain the same active ingredients as those drugs that were approved by the Food and Drug Administration, but they may be very different. The kind of binders that are there that will hold the drugs together may be different. So you've got to be a little bit careful. These are going to be allowed to be distributed after the patent has been removed. Herbal medications have not been through clinical trials. They have not been label approved. They have not had Food and Drug Administration approval. They don't have any production standards. They don't have documentation of safety and efficacy. So most of these things I would not consider to be a good drug. Now, the other thing in naming and organizing came about in the 1970s when the Controlled Substances Act organized drugs according to how likely they are for abuse potential. So they've got 5 schedules. Schedule 1 has no accepted medical use and has high abuse liability. That includes things like LSD and mescaline and marijuana. So this is where the federal government has identified something like marijuana as not being useful for medicinal purposes. It's not a drug because it hasn't been pure, it hasn't been identified for safety, and it hasn't been approved for efficacy. Schedule 2 are drugs that are available for treatments, but also have high abuse liability. And then you've got the others. Yeah.
I have a quick question. Would there be change, like, since some people are now considering marijuana to have some medical use, would that start to change where that would be classified?
Well, if they can take it through the Food and Drug Administration and say, we now know what's in here and we have done clinical trials for safety and we now have proven efficacy for such and such a disease, then it becomes a drug. Okay, so this is your homework, and I think you could read this. Basically, I want you to go find a clinical trial on something, either a drug you're interested in that's being repurposed or some new drug that is like another drug. And I want you to turn this in on the day of the first exam, because what I want you to find out about that is 2 things. 1, what the clinical trial was for, who the population was that was going to have access And I might